The focus of the lecture will be on two other membranous organelles, namely lysosomes and peroxisomes, and we'll initiate our talk about the non-membranous organelles, and we'll discuss in detail these very important non-membranous organelles, the ribosomes, which you know exist not only in eukaryotic cells, they are also organelles present in prokaryotic cells. So that's the schedule for the lecture. We'll see the basic structural again and functional features of these organelles. Let's start with lysosomes. Lysosomes were first described by Christian de Duve in 1955. He is an electron microscopist and studied the structure of the cell, the ultrastructure of the cell under electron microscope. And he is the discoverer not only of lysosomes, but also of other organelles, as you will see later in our lecture class. And because of its scientific achievements, he was awarded a Nobel Prize for Physiology or Medicine in 1974. De Duve named these organelles lysosomes because of their function. Lysis in Greek means loosening or to dissolve or to degrade, and soma is body. These are organelles in which processes of degradation occur in the cell. These are membrane-bound cell organelles which are present in all animal cells. Only terminally differentiated cells like erythrocytes, red blood cells, secondarily lack lysosomes. In early stages of their development, even red blood cells possess lysosomes in their cytoplasm. The size of lysosomes is quite variable, from 0.1 micron, which is at the boundary of the resolving power of the bright field microscope, up to 1.2 microns, which is a very large size for a cellular organelle. But on routinely stained hematoxylin using stained sections, lysosomes cannot be distinguished from the rest of the cytoplasm and other such organelles which have similar staining features. To demonstrate lysosomes in the cells, we need special stainings. One such special stain is toluidine blue. This is a section from kidneys, ren in Latin, and more specifically, this is the proximal convoluted tubule of the nephron. This tubule is aligned with simple cuboidal epithelium. These are cuboidal cells here, and you see the nucleus of one cell, that's another cell, here is one more cell with its nucleus. The cells are cuboidal. And here the toluidine blue stains the lysosomes, these purple bluish structures present in the cytoplasm are lysosomes, and these cells have abundant lysosomes in their cytoplasm. That's why we study them under microscope here. This is another picture. This is a fluorescent micrograph, an image of fluorescent microscope. The cell of the nucleus is stained blue. These red threads or granules are mitochondria. And green basically these lysosomes in this cell. And this is an electron microscope of a cell. This is a macrophage, one of the cells with abundant lysosomes in their cytoplasm, performing phagocytosis. And here you see the nucleus of this cell, and the rest of the cytoplasm is filled with these vesicles with different shape and different size and different internal composition. Some are smaller and more uniform black, others are larger with variable substances present in the lysosomes. Lysosomes are variable organelles. Their appearance depends on what is being digested in them. So the differences in the material for degradation leads to differences in how they look under microscope. So that's about lysosomes and their external, their appearance under microscope. Very frequently, to demonstrate lysosomes under microscope, we use the so-called histochemical reactions. Lysosomes contain a large number of enzymes, and for these enzymes, we have suitable substrates that, when introduced in the cells, are partially modified in the lysosomes and either precipitate there in the lysosomes or become accumulated in the lysosomes and mark the lysosomes. One such very important histochemical reaction is the reaction for a prominent enzyme in the lysosomes called acid phosphatase. It is used as a marker enzyme of lysosomes. And when this enzyme reacts with chemical substances containing lead, this lead precipitates in the lysosomes. And then, because lead is a heavy metal, when observed under electron microscope, the presence of lead atoms there accumulate electrons and make this area of lead accumulation black on the more light gray appearance of the rest of the cytoplasm. And these are two electron micrographs demonstrating lysosomes with different shape and size marked with this histochemical reaction to detect the enzyme acid phosphatase. And you can see again how variable are these organelles, from very small, tiny, these are called primary lysosomes, to large, with different internal composition based on what is being digested, secondary lysosomes. After this introduction, let's see what are the lysosomes. Lysosomes are membrane-bound organelles, like small vesicles, and inside they have more than 40 different types of hydrolytic enzymes called acid hydrolases, including enzymes capable to degrade proteins. These enzymes are called proteases. Enzymes capable to degrade nucleic acids, RNA, DNA. These enzymes are called DNases or RNases, generally nucleases degrading nucleic acids. Enzymes which can degrade carbohydrates. These are glycosidases. Different types of glycosidases for different types of carbohydrates. Enzymes degrading lipids, lipases, phospholipases, or modifying the chemicals like enzymes eliminating phosphate groups. These are phosphatases, sulfate groups from the surface of proteins and carbohydrates. These are sulfatases. So more than 40 types of enzymes. And they are called acid hydrolases because they are active in acid pH. They are only active when pH is between 4 and 5. So usually in the lysosomes, these enzymes are in inactive state because when the lysosome is formed, the pH is high in the lysosome. But later, when lysosomes become involved in degradation, the pH lowers with the help of a special, the so-called proton pump, present in the membrane of the lysosomes. This proton pump, it is an adenosine triphosphatase, actively transports protons into the lysosomes by utilizing the energy of ATP, and the more protons being pumped towards the lysosomes, here the pH changes from 6.8 to 5.5 and then to almost 4 or 5, and these enzymes become active to degrade the engulfed substances. But you might ask, when we have so many potent enzymes capable of degrading everything, how this membrane resists to these enzymes? The phospholipases, the lipases, the proteases, glycosidases, they will degrade the composition of the lysosomal membrane. Well, lysosomal membrane is rendered resistant to the proteolytic and other hydrolytic enzymes of lysosomes by modification of its composition, its chemical content. In the plasma membrane of lysosomes, there are unusual phospholipids in the form of lysobisphosphatidic acid. These are phospholipids which are not able to be degraded by these phospholipases and lipases present in the lysosomes. Also, in the in the membrane of the lysosomes, together with transport proteins, channels, and other such functionally active proteins, there are special structural lysosomal membrane proteins which are heavily glycosylated. If you remember, one of the purposes of glycosylation was protection of the protein, and these heavily glycosylated proteins are also rendered undetectable and undegradable by these proteins, by these enzymes, the potent proteases of the lysosomes. And here I give you some examples of these structural lysosomal proteins, like lysosome-associated membrane proteins, lysosomal membrane glycoproteins, and lysosomal integral membrane proteins. So that's generally the structure of lysosomes. So what is the function of lysosomes? As the name implies, lysosomes are the organelles in which processes of degradation uh, of material um, happens. Lysosomes are a meeting place where several different streams of intracellular traffic converge and deliver these substances for degradation. Basically, three different pathways are described to deliver such material for digestion in the lysosomes. Here you see a diagram showing the first mechanism, which is the mechanism of degradation of large extracellular particles, such as whole bacteria or cell debris of already dead cells or other foreign material which is being engulfed during the process of phagocytosis. And here you see the process of phagocytosis. This is the engulfment or eating of a whole bacterium. During this process, the bacterium is surrounded with a portion of the plasma membrane and then sinks into the cytoplasm. This is called a phagosome. Phagosome. The structure which is formed after the phagocytosis is a phagosome, is a phagosome. Phagosome. This phagosome belongs to the early endosome compartment, and this phagosome changes by first receiving the structural proteins for lysosomal membrane, the transport proteins, including this protein pump, which starts to pump protons towards the phagosome and acidifies the inner composition of the phagosome. When this happens, this early endosome changes into a late endosome, and then from Golgi apparatus, primary lysosomes holding inactive lysosomal enzymes come and fuse with the phagosome to change this phagosome into lysosome, or more correctly, this type of lysosome is called a phagolysosome. A phagolysosome. So that's one of the mechanisms for delivering substances to lysosomes for degradation. The second process is the so-called delivery of extracellular small particles like large proteins or plasma membrane proteins or ligand receptor complexes from the plasma membrane. This process is called receptor-mediated endocytosis and happens with the formation of clathrin-coated vesicles. These are clathrin-coated vesicles which, when formed by endocytosis, go there and become early endosomes. So this is again the early endosome compartment. And then, following the same features here, from Golgi apparatus, the early endosome receives the protective structural proteins, the transport proteins, including this proton pump, which acidifies the inner composition. And here pH is around 5.5 in the late endosomes. And then primary lysosomes containing the inactive enzymes reach the late endosomes and change this into a secondary lysosome. So that's the second route of delivery of material to the lysosomes. And the third one is for digestion of obsolete organelles or elements of the cytoplasm of the cell itself. This process is called autophagy. The cell eats, degrades its own components. Here you see an example of degradation of old mitochondria, which must be eliminated because it starts to be harmful for the cell. And initially, this mitochondrion is surrounded by a membrane formed by the vesicles of the smooth endoplasmic reticulum. And this structure, which is formed here, is called an autophagosome. 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 This is autophagosome. Then structural proteins of the plasma membrane of the lysosomes. Proton pump which acidifies the inner composition, and then primary lysosomes bringing inactive enzymes change this into a secondary lysosome, and this mitochondrion will be degraded by the enzymes of the lysosomes. So these are the three pathways for delivery of material to the lysosomes for degradation. Large extracellular substances like whole cells, bacterial cells, or viruses or cell debris, this is called phagocytosis. Small proteins, receptor-mediated endocytosis, or autophagy. The cell degrades its own unnecessary obsolete components. And now we'll discuss in slightly more detail this interesting process of autophagy. Induction of the process of autophagy is done by a cascade of signal molecules, which is very important because the cell must know which structure is not needed anymore and to degrade it. If it degrades structures that the cell needs at that moment, that's not good for the cell. And also, if the cell for some reason cannot recognize and direct these obsolete particles that are not needed anymore and degrade them, it becomes blocked and its function again suffers. And autophagy is highly regulated process. The so-called mTOR complex 1 is the main participant in this process of regulation. And when the signal comes, what is being degraded by the cell is initially surrounded by a double membrane which is produced from the smooth endoplasmic reticulum. The initial process of the formation of this wrapping structure is called nucleation, and then gradually this structure grows and encloses the autophagosome, and this process is called closure of the autophagosome. And then primary lysosomes come here and deliver the lysosomal enzymes. This is the fusion with the primary lysosomes to form a secondary lysosome, which is being degraded, or the substances in the secondary lysosome are being degraded by the digestive enzymes. And here you see one such autophagosome in which simultaneously a mitochondrion and a peroxisome, these organelles, we will discuss them shortly, are jointly being degraded by the process of autophagy. Autophagy can be divided in three well-characterized pathways. The first one is what I described till now. This is the macroautophagy. This process is for entire organelles which are being degraded in the lysosomes, and the process is not specific process. The second type of degradation is called microautophagy. It is also non-specific process in which cytoplasmic proteins are transported to the lysosomal membrane and then degraded in the lysosomes. And the last mechanism is called chaperone-mediated degradation or autophagy, and this is the only selective process. Here, special cytoplasmic proteins called chaperones recognize and bind the structures which will be degraded, and then these chaperones interact with specific receptors in the lysosomal membrane and specifically guide these proteins for degradation in the lysosome. So this is a specific process. The macro and micro autophagy are non-specific processes. Non-specific is that you don't need immediate which will help this process. You don't need receptors and surface proteins to interact with each other to guide the process. Here you need a specific protein which specifically recognizes what should be degraded in the lysosomes. This means specific and non-specific proteins process of degradation. There are different types of chaperones recognizing different targets for degradation, and each pair is specific pair. Okay. So, regardless of the fact that we have so many different degrading enzymes in the lysosomes, and these enzymes are usually quite potent, very frequently the material for degradation exceeds the degrading abilities of the enzymes. The enzymes become exhausted. And then in these cells, gradually more and more enzymes with lysosomes with exhausted enzymatic activity, and still some material for degradation present in them start to accumulate. The more long-living the cell is, the more such structures with inactive enzymes and undegraded material accumulates. These lysosomes, which have exhausted their enzymes but still have something to be degraded in them, are called residual bodies. And long-living cells like neurons and muscle cells, here you see cardiac muscle cells, have the tendency to accumulate more and more such residual bodies in their cytoplasm. And they become visible in the cells in the form of the so-called lipofuscin inclusions. When stained with hematoxylin eosin, lipofuscin stains in a very nice light brown color, and we can see these lipofuscin granules usually close to the nucleus. This is the nucleus of this cardiac muscle cell, a nucleus of another one, and you see the brownish depositions. These are residual bodies, and because this accumulates with aging of the cell, it is also called the age pigment, lipofuscin granules. So these are the lysosomes, their structure and function. Now we will briefly discuss the process of biogenesis of lysosomes, because it is a very specific process. You saw during the first lecture that lysosomal enzymes are produced by ribosomes attached to the endoplasmic reticulum, and these lysosomal enzymes initially enter into the endoplasmic reticulum. Then from endoplasmic reticulum they reach Golgi complex, and in Golgi complex these lysosomal enzymes, lysosomal hydrolases, are marked with a phosphate residue. The mannose on the oligosaccharide attached to this enzyme in the endoplasmic reticulum is phosphorylated by a special enzyme in cis Golgi network. And this process is extremely important because it marks lysosomal enzymes. Only marked lysosomal enzymes can be later packaged into primary lysosomes. If this process is prevented for some reason, lysosomes, active lysosomes cannot be formed. What happens on the next stage? These now marked lysosomal enzymes with mannose 6-phosphate travel through the Golgi complex cisterns and undergo other modifications, these that we discussed for the Golgi complex, and eventually reaches the trans-Golgi network. And here in the trans-Golgi network, there are special receptors called mannose 6-phosphate receptors. They recognize these mannose 6-phosphate residue on the surface of the lysosomal enzymes. And such receptors are located in regions of the trans-Golgi network membrane, and these acid hydrolases marked with mannose 6-phosphate become specifically attached to these receptors and concentrated in specific regions of trans-Golgi network. Then, from trans-Golgi network, cluttering-coated vesicles, but which contain large concentrations of lysosomal enzymes. Very rapidly, after the budding of cluttering-coated vesicles, the cluttering coat is shedded off the surface of these vesicles, and these structures which are formed are called primary lysosomes. They contain inactive lysosomal enzymes. Here, in the primary lysosome, the pH is high. The pH is between 6.5 and 6.7. At this pH, the enzymes tend to be tightly attached to the receptor and also rendered inactive. Then this primary lysosome reaches the endosome compartment in which material for degradation is already present, and these primary lysosomes fuse with the endosomes, and here in the endosomes, because the proton pump is present, the pH is lower than 6.5. It is around 5, and at this pH, around 5, lysosomal enzymes tend to dissociate from the receptor and become free here, and then additionally the acidic conditions activate them, and now this structure is called a secondary lysosome. You can see that receptors are utilized again, reused from the endosomes, receptors, but in the form of vesicles. These are cluttering-coated vesicles which travel backwards to the Golgi complex, and these receptors can be utilized in many cycles of transportation of lysosomal enzymes to the secondary lysosomes for degradation. So that's how lysosomes are being formed, and we have primary and secondary lysosomes. Based on what is degraded in the secondary lysosomes, they can be autolysosomes, phagolysosomes, or crinolysosomes. Crinolysosomes can be seen in secretory cells when our body does not need the specific secretion of this cell. Some of the secretory vesicles fuse with the late endosomes, and their content is degraded into secondary lysosomes called crinolysosomes. In medical genetics and in pediatrics and in neurology and in internal diseases and pathology, you will discuss a large group of so-called lysosomal storage diseases. These diseases are due to the genetic defect, usually, in the genes for some of the enzymes, many enzymes present in the lysosomes. Depending on which enzyme is defective, will have different symptoms in these individuals. And here you see some of the most frequently seen such lysosomal storage disease, like the syndrome of mucopolysaccharidosis type 1. This is the faulty enzyme, and because this enzyme cannot degrade different carbohydrates, these carbohydrates accumulate in the extracellular matrix and inside the cell, damaging the cells, predominantly the cells in muscles, bones, and cartilages, and also neurons in the nervous system. Another such frequently seen genetic disorder is the McArdle syndrome. That's the enzyme which is defective. Here, the main symptoms are from the skeletal muscles. A very famous disease, especially in North America, in Canada, and also in Europe, but not so frequent there, especially in individuals with Jewish ancestors in the family, is the Tay-Sachs disease. Here, metabolism of lipids is obstructed, and the main symptoms come from the nervous system because neurons become affected. And here you see a disease which is called eye cell disease, or this is the disease of empty lysosomes. Here, the enzyme marking the lysosomal enzymes is defective and is not active. So lysosomal enzymes cannot be marked with phosphate residue on their manuals. And because they cannot be marked, they cannot be recognized in Golgi complex to be included in the primary lysosomes. And Golgi complex produces some empty lysosomes, which lack enzymes in them. So lysosomes are not really formed. And this is an eye cell disease. Eye comes from inclusion cell disease. So these are only a few of the many lysosomal storage disorders. Every enzyme that is defective leads to a specific disease. So you will discuss these diseases in detail in medical genetics and pediatrics, mostly because these diseases are genetic and symptoms appear early in postnatal life. Okay, so these were lysosomes. Do you have any questions about lysosomes? Okay, so now we'll move to another intracellular structure in which degradation of damaged or unneeded proteins happens. See, only proteins. The major place for protein degradation in the cell are lysosomes, but the cell possesses another mechanism to which it can degrade unnecessary damaged proteins. This is not an organelle, this is a large enzymatic complex present in the cytoplasm. We will discuss it now because its function is related to the function of lysosomes, and I want just to point about another structure which serves similar function to lysosomes. This protein complex is so important that usually it constitutes nearly 1% of the cell protein content. Proteasome mediates degradation of abnormal proteins, proteins which are incorrect, misfolded, denatured, or of normal proteins which must have a very short life. They fulfill rapidly their function and then they must be rapidly eliminated from the cell. Such proteins are involved in the regulation of the cell cycle and cell division. These are the mitotic cyclins, which are rapidly synthesized, act, fulfill their duties, and then they are being also rapidly degraded in the proteasome. Also, transcriptional factors and tumor suppressors are being degraded by the proteasome. And what is this proteasome? Each proteasome consists of a hollow cylinder, you see it here, this structure, which is called the core particle, and two, in purple, regulatory subunits or particles. Proteasome is capable to degrading only proteins which are marked for degradation. So this protein cannot be degraded by the proteasome unless it is marked so that the proteasome can recognize it as protein due for degradation. The process of marking of the proteins is called ubiquitination of the protein. Why? Because proteins targeted for degradation are marked by the addition of small proteins called ubiquitins. Usually, several ubiquitin molecules should be added to the protein so that the protein can be recognized by the proteasome. Usually only one ubiquitin is not enough. More than 3, 4, 5, 6, even 8 ubiquitins are needed so that the protein can be recognized by the proteasome for degradation. And this regulatory subunit recognizes the ubiquitin tag. The protein comes here, binds with the regulatory subunit with its ubiquitin tag, and this regulatory subunit has also enzymatic activity. It removes ubiquitin. It has fulfilled its duties, and then this ubiquitin recycles again in the cytoplasm to be utilized in another cycle of protein degradation. And then this regulatory subunit guides the protein towards the core particle. The core particle is a protease complex, which degrades the protein. It cuts the polypeptide bonds between amino acids and produces amino acids or short oligopeptides, which are gradually liberated by the secondary regulatory particle. And that's how proteins are being degraded in the proteasome. Remember this proteasome. The disruption of the normal functioning of the proteasome leads to accumulation of unneeded proteins in the cell, and it is a major factor in pathogenesis of many degenerative disorders and diseases that you will study in neurology, in pathology. And now it is time to discuss another membranous organelle in the cell, namely these are the peroxisomes. Peroxisomes were also identified by Christian de Dieu in 1967. They are called peroxisomes from Greek word peroxide and soma body. Why? Because in these organelles, the chemical reactions of peroxidation are happening. These organelles can be with a small size from 0.5 to from 0.1 to 0.3 microns, and then they are called microperoxisomes. And most of the peroxisomes in human cells are with this diameter. But sometimes we can see larger peroxisomes, larger than 0.5 microns, up to 3 microns, and these peroxisomes are called macroperoxisomes. These are spherical organelles, and usually one eukaryotic cell has between 70 and 100 peroxisomes. Of course, there are cells with more abundant peroxisomes, like liver cells, but on average, each cell has between 70 and 100 peroxisomes per cell. In most of the animal cells, peroxisomes have a single layered membrane, an inner composition called matrix, and in the matrix we can see a dense crystalloid core which is called nucleoid. But human peroxisomes lack this nucleoid. It is not present in the human peroxisome. So you can judge here that these electron micrographs are from animal cells, not human cells, because they have this crystalloid or nucleoid in their matrix. Here, the nucleus is stained blue, mitochondria are red, and the green spots are peroxisomes in this cell observed under fluorescent microscope. What is the structure and the function of peroxisomes? As I said, they have a single layer membrane, similar to the plasma membrane, and matrix. This matrix contains more than 50 different types of enzymes. The most abundant enzyme, which constitutes around 40% of its protein content, is the enzyme catalase. So that's the dominant enzyme in peroxisomes. Peroxisomes are also involved in the process of beta oxidation of fatty acids with a very long chain, more than 20 carbon atoms. These very long fatty acids cannot be degraded by the mitochondria. Their degradation starts in peroxisomes and it is completed in mitochondria. So the second set of enzymes are the enzymes called beta oxidase. This enzyme is involved in the degradation of very long fatty acids. Also, in peroxisomes, we have amino oxidases. Amino oxidases are enzymes degrading amino acids. And not in humans, but in animal peroxisomes, we have urate oxidase. This urate oxidase has tendency to crystallize and form the nucleoid or the crystalloid core. Because this enzyme is not present in the human peroxisomes, we don't have the nucleoid there. What are the functions of peroxisomes? I already mentioned them. This is beta oxidation of very long fatty acids and also different processes of oxidation of amino acids, of toxic, harmful substances for the cell. Generally, these oxidative enzymes within the peroxisomes utilize molecular oxygen to remove hydrogen atoms from specific organic substances. This is the oxidative reaction. They oxidize the organic compound and produce hydrogen peroxide, which is itself toxic. But this enzyme catalase, which is the most abundant enzyme in peroxisome, can use this hydrogen peroxide to either oxidize other organic substances by inactivating them and producing water, or to inactivate hydrogen peroxide itself to produce water and molecular oxygen, which is these reactions. So that's the function of peroxisomes, reactions of oxidation or peroxidation. And about 25% of the ethanol we drink is oxidized here in peroxisomes to acetylaldehyde by these reactions. So peroxisomes are involved in detoxification of different toxic substances by oxidizing them. So peroxisomes are very special organelles. They can be derived from the endoplasmic reticulum, and they can also replicate by fission like mitochondria. When they are produced de novo from the endoplasmic reticulum, initially from endoplasmic reticulum, empty peroxisomes are produced. These empty vesicles contain specific receptors on their surface which recognize the enzymes for peroxisomes which are produced by free ribosomes in the cytoplasm. And then actively these enzymes are delivered to peroxisomes and new peroxisomes are being formed in the cells. So these are the peroxisomes. Any questions about peroxisomes? Any questions? Okay, so now we move towards the last organelle for today. This organelle belongs to non-membranous organelles, and these are the ribosomes. Ribosomes were first observed in the mid-1950s by a Romanian biologist called George Palade by using electron microscope. George Palade, together with de Duve and some other electron microscopists, were awarded a Nobel Prize for the study of the structure of the cell in 1974. The term ribosome was proposed by another scientist, Richard Roberts, in 1958. Here you see George Palade and his electron microscope, and that's an original electron micrograph he made, and these are the cisterns of endoplasmic reticulum, and you see ribosomes which are attached to the surface of the endoplasmic reticulum, and this is rough endoplasmic reticulum, but you also see free ribosomes in the cytoplasm. So ribosomes can exist in two forms, either free or bound to the surface of the endoplasmic reticulum in the form of rough endoplasmic reticulum. Ribosomes are the organelles which are guiding the process of protein synthesis. This process is called translation. The synthesis of protein is starting when messenger RNA is produced in the nucleus. So that's how the genetic code from DNA is transcribed into the genetic code of RNA. This molecule, because it contains the uh, information for one polypeptide chain, is called a messenger RNA, mRNA. And then this messenger RNA is transported via the nuclear envelope in the cytoplasm. And then when it reaches the cytoplasm, it binds with the ribosomes, which are guiding the process of translation. And we need ribosomes for the process of translation because they ensure accuracy for this process. With the help of ribosomes, usually one mistake is made for every 10,000 added amino acids. Usually proteins are having a length from several hundreds to one or so thousand of amino acids. Rarely we have polypeptide chains longer than 2000 amino acids. So you can see one mistake for every 10,000 amino acids is a very huge accuracy. But yet sometimes abnormal proteins can be produced. That's why we have the proteasome and this machinery which recognizes them and degrades them. Ribosomes as organelles, here you see many ribosomes bound to a single molecule of RNA, and the same is here. This is the initial process, and here the protein translation has efficiently performed, and here you see from every ribosome a newly synthesized protein appears. So ribosomes are a complex catalytic machine which is made of more than 50 different proteins called ribosomal proteins and several special RNA molecules which are called ribosomal RNAs. These are the ribosomes. You will ask me why there are two different types of ribosomes, because this is the bacterial ribosome, the ribosomes of prokaryotic cells, and here we have the eukaryotic ribosome. I hope you remember that both eukaryotic and prokaryotic cells have ribosomes. And these ribosomes are very close but not identical in their structure. The bacterial ribosomes are smaller than the eukaryotic ribosomes. The eukaryotic ribosomes are 80 Svedberg units, while the prokaryotic ribosomes are 70 Svedberg units. Regardless of this fact that they have some structural differences, generally prokaryotic and eukaryotic ribosomes share similar structure. Each ribosome consists of two subunits, one which is called a large subunit and the other which is called a small subunit. These subunits exist separate in the cytoplasm and they assemble to form a real ribosome only during the process of translation. When the translation ends, immediately the two subunits separate from each other and ribosomes are not existing. If we see ribosomes, they are only present during the process of translation of proteins. Large ribosomal subunit of eukaryotic ribosomes are with 60 Svedberg units their mass. The small subunit is 40S or 40 Svedberg units. For prokaryotic ribosomes, the large subunit is 50S, the small subunit is 30S. You don't have to memorize the molecular weight, you won't be a molecular biologist, but you must know these Svedberg units or the mass of the whole ribosome and individually the large and small subunit. And let's see what is the composition of these large and small subunits. As I told you, ribosomes consist of proteins and ribosomal RNA. And you see, the small subunit has around 30 proteins and one molecule of ribosomal RNA. It has 1,900 nucleotides. It's not necessary to remember the exact length of the ribosomal RNA here, but if you wish an excellent mark, you must know the Svedberg units 18S. This is what molecular biology uses in the description of the ribosomes more frequently than the exact length. The large subunit has around 50 proteins and 3 molecules of RNA, 2 short of 120 and 160 nucleotides, 5S and 5.8S, and 1 large RNA of 4,700 nucleotides or 20S ribosomal RNA, Svedberg units. So this is the composition of the eukaryotic large and small subunit of the ribosome. What about the large and small subunit of the prokaryotic ribosome? Again, the small subunit contains a single RNA, ribosomal RNA, slightly shorter from the eukaryotic one, 1,540 nucleotides or 16S and around 20 proteins. Large subunit, around 30 proteins and only 2 ribosomal RNAs. One long, 2,900 nucleotides, 23S, and one like the smallest of the 3 ribosomal RNAs in eukaryotic large subunit, 5S, or again 120 nucleotides. So that's the general structure of the ribosomes, both prokaryotic and eukaryotic ribosomes. So how these ribosomes guide the process of translation? To guide the translation, ribosomal subunits contain binding sites. There are at least 4 binding sites for RNA molecules present in each of the 2 subunits. One is for binding of messenger RNA, and this binding site is present on the small ribosomal subunit. This dark green portion is the small ribosomal subunit, and this is the messenger RNA binding site. Then both small and large subunit have 3 additional binding sites. One is called A site, the other is called P site, and then an E site. These binding sites are for binding transport RNAs. Transport RNAs are transport molecules which deliver amino acids during the process of protein translation. And you can see that when the 2 subunits join together to form the ribosome, this is the small and large subunit, when they join, their corresponding sites complete each other to form the functionally active A, P, and E site of the ribosome. What about protein translation? The protein translation begins when messenger RNA comes from the nucleus into the cytoplasm. And in the cytoplasm, we have many small and large ribosomal subunits, of course, detached from each other but present in the cytoplasm. The small ribosomal subunit binds a section of RNA initiator transport RNA, which is attached to the amino acid methionine. Every protein starts with this amino acid at its beginning, and this initiator transport RNA holding methionine becomes attached to the P side of the ribosomal, small ribosomal subunit. And then this small ribosomal subunit binds with messenger RNA and starts to scan the nucleotides of the messenger RNA until it reaches the so-called start codon, AUG. This start codon encodes the addition of the amino acid methionine. And here in the transport RNA, we have C, we have, we have U, A, C as anticodon to the AUG codon, and hydrogen bonds are being formed here, 2 for adenine and uracil, and 3 for guanine and cytosine, and this complex becomes firmly attached here. And then the next codon, which encodes for the next amino acid, for example, it might be ACG, asks for another transport RNA holding the complementary anticodon and bringing the next amino acid. And that's what happens on the next stage. The next transport RNA, which specifically binds with the codon after the start codon comes, it becomes attached firmly by hydrogen bonds to the codon, and then there is a special enzyme in the large subunit called peptidyltransferase, which forms a peptide bond between methionine and the next amino acid. Then the small subunit moves 3 nucleotides further along the messenger RNA, and when this happens, the first transport RNA is now located in the E site. The second transport RNA is located in the P site, and it contains the growing protein, and the A site, which is the accepting site, is free again to receive the next transport RNA. Next peptide bond is formed, and this process continues and continues until the ribosome reaches the so-called stop codon. Stop codon can be one of these 3 codons, UAA, UAG, and UGA. These codons, they do not encode the addition of amino acids, but they are recognized by a special terminating factor which binds to this codon and prevents further addition of amino acids and signals for the end of the protein synthesis. When this happens, the newly synthesized protein is liberated, the 2 subunits are disintegrated, and protein synthesis ends for this ribosome. Then it can be utilized in another process of translation again and again. So these are the specificities of the translation process with its initiation, elongation, and termination. So that's everything about ribosomes for today. We will finish a little bit earlier today. As I lecture week we in the lecture hall on the theory of the on the territory of the university, and the topics of the lectures next week will be elements of cytoskeleton. These structures belong also to the non-membranous organelles, and you will discuss also the structure of the nucleus, the interface nucleus. So this will be topics of the lectures for the next week. Okay, thank you for your attention, and I hope next week we'll meet in presence in the university.